Right, ladies and gentlemen, it's lovely to be here again, and I'm bringing, I suppose, fraternal greetings from the Glasgow Natural History Society. I'm wearing the t-shirt tonight for that. And it's interesting, we have rather similar issues over publications and excursions and all kinds of things. We put our journal is now available online through the Natural History Heritage Library, run from the United States. So all our issues going back to the very beginning are there. So if you've not really thought about doing that, it's a free service and something you could do if you haven't considered it yet. Anyway, so I'm here, as some of you learned earlier, with a group of undergraduates and postgraduates and staff from Strathclyde University in Glasgow, University of Glasgow in Glasgow, and the University of Edinburgh. So we're a combined group this year, and we're staying up at Simla and doing research on marine turtles, frogs, and what else? Geography. Geography, yes, we've got a real team of geographers this year, and it'll be interesting to see what they get up to. So, I should first of all give some acknowledgements. This is work I'm reporting from almost 20 years of research we've done on the stream frogs of Trinidad and Tobago. And the collaborators that I should mention are Suzanne Livingston, Michael Jowers, Joanna Smith, Alex Ryan, Mark Greener, Ruth Shepherd, Chris Pollock, and Emily Hackenden. Emily's work, which I'll report on, was done just last year. So lots of people helped fund our work here, but I'm not going to list them. Oops. No, have I pressed the wrong thing? Right, names. One of the problems about working on any kind of organism, I think, in the tropics, particularly the tropics, is that the names keep changing. Common names may not change too much, but the scientific species names keep changing because of taxonomic revisions. And this can cause a lot of confusion. It's bad enough having to learn one complicated Latin name, never mind 3 or 4 for the same organism. So just to kind of review what the species I'm talking about are actually called, when Jake Kenny did his Amphibia of Trinidad, which I'm sure many of you will have looked at, he noted that the stream frogs, I'll call them that you get in Trinidad had been called Prostherapis trinitatis, but he reckoned that they should be in the genus Phyllobates, and they belonged to the huge neotropical family of dendrobatids, the poison frogs. However, Lamarca in Venezuela, after doing extensive work on these frogs, realized that there was a special group which really ought not to be lumped together with the dendrobatids, and he gave them the name Mannophryne for the genus, and a new family name, the Aromobatidae. And we can now say that there are 20 described species of Aromobatids. They occur in eastern Venezuela, Trinidad and Tobago. They don't seem to occur so far anywhere else. I think the number of species is provisional, as it often is, because some of the species boundaries are not yet clear. Now, Jack Kenny did not mention a population in Tobago, which I've never been terribly sure why. Did he know that there was a similar frog in Tobago, or had he not bothered to visit Tobago often enough to check? Anyway, it wasn't until Jerry Hardy in 1983 named the Tobago species as Almani, so Mannophryne almani, and it was later confirmed to be a distinct species. It wasn't the same as the Trinidad species. So we have 2 species in the Republic of Trinidad and Tobago. Now, what are the characteristics of these frogs? In some ways, they are similar to dendrobatids. So, mostly they are active during the day, which of course is distinct from most amphibians, which are night active mainly. They're also distinctive in laying their eggs on land, not in water. And all species, as far as we know, have parental care. In other words, they look after the eggs after they've been laid in some ways. And in Mannophryne, the parental care is always by the males. So it's the males that look after the eggs. In some dendromatids, it's the female or the male, and in some cases even by both. And another thing that's characteristic of Mannophryne, similar to dendromatids, is that once the eggs hatch, they are transported by one of the caring parents. In Mannophryne, always by the male. So they're carried about for a time. We'll talk about that later. However, there are some differences between the aromabatids and the dendromatids, which is why Lamarck has set up this new group. So Mannophryne lack toxins. So to call them poison frogs is pretty stupid because they're not poisonous. Unfortunately, a new major work has come out just in the last week or two, trying to give good common names for the species in the Caribbean region of amphibians, and unfortunately they have kept to the old convention of calling these poison frogs. I think it's daft. John Murphy agrees with me that it's daft, but they've done it. And the other thing about them that's special, that separates from the dendromatids, is that the behaviour of Mannophryne and its coloration is cryptic. It's not obvious. So the dendromatids are generally very brightly coloured. They're obvious. It's a warning that they're toxic. Whereas Mannophryne are cryptic. They're brownish. They fit in with the background very easily. They don't warn you that there's something dangerous about them, because there isn't anything dangerous about them. So the themes of this talk, having sorted out the names, are first of all to say a bit about their distribution and status, conservation status in Trinidad and Tobago, a bit about tadpole transport and habitat selection, a bit about female territoriality and throat colour, a bit about male calling and colour change, make a little comment about other Mannophryne species, and then briefly discuss what we're trying to do this year and other things that really need to be sorted out about these species. Now, Brenmar, you better warn me if I'm going on too long. We started a bit later than I expected us to. Okay, gone too far. That's it. So, distribution and conservation status. So, the Trinidad stream frog, Mannophryne trinitatis, Kenny described it as occurring on the margins of steep streams, intermittent streams throughout the northern range, and also an isolated population in Tamina Cave in the central range. Michael Jowers did a little bit more on the distribution, confirming that it was very widespread throughout the northern range, except where there had been severe deforestation. It's also found in the central range more extensively than Kenny reported, so you get it from Mount Harris in the east to Grand Couva in the west, including Tamina. Michael Jowers never found any in the south. He looked quite hard in the Trinity Hills area, but couldn't find any Mannophryne there. However, a more recent report by Mohammed and et al. In Living World reported finding Mannophryne trinitatis in a pond on Edward Trace. Brenmar and I are hoping to go down there and confirm or not confirm that observation in the next week or so. But it does need confirmation because it would be the first properly authenticated report of it outside those two original areas. The IUCN Red List for most recently 2010 regards Mannophryne trinitatis as vulnerable and declining in numbers, and the criteria for that are its occurrence over a relatively small area. Well, obviously if it only occurs in Trinidad that would be relatively small, and they say that there's considerable decline in the extent and quality of its habitat. An additional problem is that there had been a report of chytrid fungus infection in these frogs. Chytrid, as many of you will know, is one of the things that's responsible for the widespread decline all over the world in frog populations. It's a very severe, or can be a very severe infection. However, there had been no actual published field assessments of this frog. IUCN's listing as vulnerable was based on a committee decision where they said, oh, we think its habitat is declining and therefore it must be vulnerable. So what we did was we did some actual surveys. These were quite extensive over a few years. We listened to them calling, we looked at where we could see them, and we also carried out what we call removal sampling, which is a bit more invasive for these frogs. In other words, we took a couple of small stream valleys and went up the valley one morning, collected every frog we could see, put them in an aquarium tank for later, and then did the same the next day in the same stream. And we did that for 3.5 days or so until we couldn't find any more frogs, or the ones that were left we could see and knew how many there were. And Mark, who did this, was really astounded by the results because his reckoning from the visual encounters and from the calling that he heard was that there might be about 50 frogs in a stream. Removal sampling showed the number to be nearer 300. In other words, there are a lot of these frogs. We also reassessed the chytrid story, and our attempts to discover whether there was any chytrid or not found none in this species or any other species in Trinidad. That doesn't mean to say that the previous report was wrong. It may well be that it is correct and that the fungus has died out. The previous report by Mohammed et al., by Elemu et al., basically said that there was chytrid there at a fairly low level, but it wasn't causing any illness in the frogs. They didn't seem to be dying off from it. So we didn't find any at all. So our conclusion, based on the kind of huge numbers of frogs that we estimate occur in the northern range, a guesstimate of about 3 million, also add on the Tamina sample, we reckon that this frog is not vulnerable to extinction. It's in a fairly healthy condition, but it does need to be monitored because if the habitat declines, then it'll decline too. And as we've heard, there are problems with forest fires in the northern range, and climate change and problems with the water supply in the northern range may well cause difficulties for this species. We also have looked at the Tobago stream frog. Jerry Hardy found it in the streams of northeast Tobago, similar habitat to in Trinidad. Rick Lettinen a few years ago did some further work on it and extended its range into the south, not very far south, but a bit further south and west of Tobago. And on the basis of his work, which he published, the IUCN moved it from critically endangered to vulnerable. So it's still a vulnerable species. And I think that's reasonable because its numbers, it's not as densely populated, and Tobago is a much smaller island, and therefore its range is pretty small. Again, chytrid was detected. We reassessed it and found none. But our sample size was quite small. Mannophryne almanae is a difficult frog to catch. And we only managed to assess 31 of them, but no chytrid.
How did that compare to others?
They had a slightly larger sample.
Oh, okay.
Yeah, not hugely large.
Were you able to kind of replicate to
Some of the sites would say yes. Not all, but we've had some different sites, but some would say so. Okay, let's go on to some of the more kind of, to me anyway, interesting biology that we've tried to do on these frogs. One of the things that you find is that The frogs are very, very common in the northern range, as I've mentioned. I'm sure many of you, when you've been travelling up and down the roads there, will come round a bend and there's a little stream and hear, and it's all Mannophryne trinitatis. Huge numbers, very abundant. When you start to look for the tadpoles, however, things are a bit more tricky because most of the streams are full of predatory fish. Lots of guppies, but more particularly lots of rivulets. And you just don't see tadpoles. So where are they? What's happening after the eggs hatch? The males put them on their back, or tadpoles jump on the back, and off they go, looking for a stream. The streams are full of predators. So we were a bit puzzled by this for some time. For a few years I collected tadpoles from Tamina Cave, where there are no predatory fish, and that was the only place I could find them. And then, in the Lochinau Valley one year, we came across a stream where the stream goes in a culvert under the road, and then down a cliff. And the cliff, of course, acts as a natural barrier to the fish. Now, rivulus is a pretty smart fish and is able to move over land under certain circumstances, but that cliff would provide a real barrier even to rivulus. And we found a pool there, and we thought, my goodness, there are a lot of tadpoles here. They were all Mannophryne trinitatis, and we counted them. The student who did this didn't like the job, but they did manage to count all the tadpoles, and they found over 900 tadpoles in one pool. Now, if this had been a cane toad spawning, 900 would be quite a small number. But remember that the clutch size for Mannophryne trinitatis is about 12. So 900 tadpoles implies that something like 75 males from all over the place, because they're not clustered, they're distributed widely, about 75 males must have come to this pool and deposited their tadpoles in there. And that set up a kind of working hypothesis for us, which was that transporting males are very picky about where they deposit their tadpoles. And they spend quite a lot of time and effort looking for a good spot. And the good spot is defined by lacking predators. So that was our working hypothesis. Could we get evidence to support it? Well, rather, I didn't think this would work actually, but what we found was we could capture transporting males, bring them back to the lab, and then set up a choice experiment for them. To ask them to tell us which kind of pool they liked to deposit the tadpoles in. So we set up long tanks in our laboratory at the University of West Indies, and at one end of the tank there was a nice pool of clean water with no predators in it, and at the other end of the tank there was a predator, a caged predator. So we weren't doing anything unethical. We weren't going to be killing off these tadpoles by exposing them to actual predators, but the predators were visible and chemically detectable. So we asked ourselves, right, where will the frogs deposit their tadpoles? And it's the only time I've ever got a 100% result. All the frogs deposited all their tadpoles in the predator-free pools. It was perfect. So it was clear that they were choosing. And notice how long it takes them to do this. I'll not bother you too much with the difference between southern slopes and north coast populations, but we did think there might be some difference in the way they behaved. And the southern ones took a little bit longer to deposit their tadpoles than the north coast ones. So we thought, right, let's give them a harder choice. Let's set up an experiment where both pools have got predators. What do they do then? Well, what we found was that they didn't deposit their tadpoles straight away. It took them ages to deposit the tadpoles. They sat around, they wandered about the tank. A few of them eventually deposited their tadpoles in the predator-containing pools, but most of them deposited their tadpoles on the damp leaf litter at the bottom of the tank, rather than expose them to the predators. So it was wet, but it wasn't a pool. But that was better than leaving them to the mercies of any of the predators. And we then did a field experiment, which was, again, looking to see where they might deposit their tadpoles. So this is a sort of rough sketch map of the stream at Mount St Benedict. Heard Mount St Benedict mentioned earlier today already. And what we did was, all those numbers represent places where we put little tubs of clean water up to about 25 metres away from the stream. And our question there was, would the migrating frogs carrying their tadpoles deposit their tadpoles in these artificial pools that we just set out? In other words, really we're saying they're looking for a water source. It doesn't matter if it's an artificial one, if it's a new one, that's what they're looking for. And we found many of these tubs very soon had tadpoles in them. And what was really interesting was most of them only had 1 or 2 or maybe 3 tadpoles in them. So our conclusion was that the transporting males were, in a sense, hedging their bets. They didn't put all their tadpoles in one place. They distributed them amongst a number of places, presumably with the intention that that would increase their chances of survival and getting through to metamorphosis. The next question we asked about this was, if you're a male transporting your tadpoles, obviously your tadpoles are a big investment. They're your future. They're you passing on your genes to the next generation and all that. And therefore they're important to you. But if it takes you a long time to find a suitable pool, isn't that going to be rather dangerous? Possibly to the tadpoles. They're on your back, after all. They're not they're not in a stream. Will they dehydrate? How will they survive that sort of relatively long time? And what about you as the transporting male? Wouldn't having a load of tadpoles on your back slow you down, make you more vulnerable to predators? The longer that it happens, are you able to feed while you're transporting your tadpoles? So we asked those kind of questions. And the first thing we did was to collect some newly hatched tadpoles and keep them in our laboratory on a damp surface without any food for several days, asking the question, do they decline in quality, or are they still good tadpoles at the end of that period? If you then put them in water, give them some food, do they grow as well as freshly hatched tadpoles? And they did. There was no sign of deterioration over a period of 4 days, which was roughly the time that we thought the tadpoles, the males, might take to find a place. And we also asked then about the transporting males. We did some jumping experiments with them. Category of frog, females, calling males, transporting males, and the distance they were able to jump when we stimulated them with a short stick. You'll notice that the females jump longest of all those 3 categories, but the calling males and the transporting males are indistinguishable in the distance they're able to jump. And that's despite the fact that a full load of tadpoles is somewhere around 20% of the frog's body weight. And you'd think, surely, if you had 20% of your body weight on your back, would you be able to run as fast as without it on your back? And I think the answer is no. But these frogs seem to cope with that load without any obvious problem. We also found that they are feeding during the transporting phase. So it's not costing them in the sense of not being able to forage for fruit flies and other things that they eat. So our general conclusion from all that set of work was transporting males go to a great deal of care to avoid depositing tadpoles in predator-containing pools, and the transport period incurs no obvious costs either to the tadpoles or to the transporting males. And I'm sure there's more things that could be discovered about that, but we've not gone further into that particular question. Next thing I want to talk about, and this will be the main topic, but I'll try to keep it short because it's a little bit late. Sexual dichromatism in frogs, with Mannophryne as a case history. Now, sexual dichromatism, for those who are not familiar with the term, means males and females being coloured differently. So, if you think about the way that amphibians, particularly frogs, attract a mate, everyone knows that frogs, particularly males, call. They make various kinds of sounds. You all have heard the little frogs that you get in the ditches all over Trinidad going boom, 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 that kind of noise at this time of year. And those are males calling to attract a female. So we know that sexual signalling in frogs is very largely acoustic. It's through sounds that they make. And that links well to the fact that most amphibians are essentially nocturnal. They're active at night, where visual signals you'd expect would be not so effective. How are you going to signal to someone when it's all dark by a visual signal? But, rather surprisingly, there is increasing evidence that visual signals are used by frogs. And these signals can involve colour changes, they can involve arm waving, they can involve vibrations of the throat, which are detected visually, and these occur both in nocturnal and diurnal species. And a review just a few years ago by Bell and Zamudio just kind of talked about the different kinds of visual signalling in frogs and classified sexual dichromatism into two kinds. There was ontogenetic, in other words, it's a developmental difference, where permanent differences between males and females appear at some stage in development. And then there's dynamic sexual dichromatism, where the colour difference is temporary, it's usually in males, and it's related to mate attraction. And in their review, one of the species they mentioned was Mannophryne trinitatis, where the males turn black when they're calling. And if you've ever looked carefully at those little frogs in the streams and looked to see what colour the males are that are calling, you'll see that they are black. However, I think Bell and Zamudio hadn't done their reviewing of the literature entirely perfectly, because Kentwood Wells, who was the first person to study the behaviour of Mannophryne trinitatis in detail, had noted that the females have a distinct colour difference from the males. They've got very bright yellow throats with a little black band called a collar posterior to the yellow throat, and that doesn't occur in the males. So Mannophryne trinitatis, from the literature, has two kinds of sexual dichromatism. And Wells also noted that the females show territorial behaviour. Now this is quite unusual in frogs. If there's any territorial behaviour, it's usually the males, but in this frog, the females defend the territory against other females. And they just visually do it by displaying their throats and vibrating them. So the signal seems to be the yellow throat of the female. And just another point to make before we go into detail of what we saw, yellow in many animals, lots of birds, lots of other kinds of animals, is a signal of quality. It's often used as a signal that you are a high quality, good individual, fit to breed with. And there's evidence of a relationship between the role of yellow colour based on carotenoids with immune system function and other aspects of quality of individuals. So, what have we done? We've just submitted this paper. I have no idea whether it will be published or not. But our first question was, is the yellow throat in females a case of ontogenetic dichromatism? So what we did was we collected some tadpoles of Mannophryne trinitatis, we reared them through to metamorphosis, and then reared them through to the juvenile period, to nearly adulthood, and photographed their throat colours to see if the yellow colour appeared with time. And we found that the dark collars occurred in both males and females, but very variably. The throat colour in all individuals starts as grey or white, but at about 7 weeks, some of the individuals begin to show pale yellow colour. So the ones on the left, down that figure there, are all early stage ones. On the right are later stage ones. And if the yellow colour started to appear at a fairly early stage, it got stronger. But in individuals that didn't get any yellow colour at that sort of stage, it never appeared. So our conclusion was that in the males, they stay grey to whitish, and in the females, the yellow colour develops with time. So it is an ontogenetic development, change and difference. We then asked ourselves, how variable is the throat colour between individual females? And Mark Greener, who collected lots and lots of these frogs, photographed lots and lots of frogs. The sample size was about 500, and he photographed the throats and then measured their degree of yellowness using the red-green ratio, using fancy techniques for measuring colour. And he found considerable variation in the the red-green ratio of the yellow patch. You can see here a bunch of females, some of which have got almost orange throats, some of them are a bit paler, some of them are very pale. The collar, as you can see, is variable in its darkness. Sometimes it's always black, sometimes grey, sometimes a wide band, sometimes narrower. And you'll notice also that in a few individuals, the yellow colour extends posterior to the collar. So our general conclusion was there is a great deal of variability in the yellow colour. It's not just the yellow colour that varies, but clearly females vary considerably in this signal of the throat. So, have you got any idea whether or not it's true that the colour of the throat is a signal of quality? Well, we thought, first of all, okay, so this is a signal where females are showing territorial behaviour. So, if we follow a couple of females having a fight about a territory, and then we record which is the winner and which is the loser, one that's chased away and the one that stays victorious, and then catch them, and then record their throat colours, this would give us extremely good evidence as to whether or not this was true. I don't know if anyone's ever tried to catch Mannophryne trinitatis. Some people here have and find it already quite difficult. And if you're trying to catch two particular frogs which have just displayed a particular behaviour, forget it. These streams, as you'll know, I'm sure if you've walked through the Northern Range at all, are kind of steep, tumble-down streams with big rocks, little overhangs. The frogs are extremely proficient at hiding whenever anyone approaches. So the idea that we could watch a territorial encounter and then catch the two combatants was something that just wasn't going to work. So our second idea, and biologists are always doing this, if they can't find a direct measure of what they wanted to find, they use a substitute, which you might call a proxy for quality. So our idea was we might catch a bundle of these females and assess some other aspect of their quality that might correlate with this idea of yellow colour. And the one that we chose was their escape responses. Now, we'd done quite a bit of work on escape responses in these frogs already, so we knew that we could get them into an arena, gently stimulate them, they would jump, and we could measure the jumps. And so what we did was we took quite a large sample, 81 females, this is what Emily did last year, and she photographed their throats, and she measured their escape responses in the arena. I'm not going to go into all the detailed results here, but what she found was a strong positive relationship between the red-green ratio, the yellow colour, and size. So bigger frogs had brighter, more orangey coloured throats. But also she found that there was a strong positive relationship between the red-green ratio and two escape measures. Maximum distance in a single jump and total distance travelled before stopping. And we reckoned that those were quite reasonable proxies for quality. That, if you're going to be a good quality frog, you need to be able to escape from predators, and the better you do it, the better the chances are you have of escaping. So we found that total distance jumped, maximum distance related positively to the red-green ratio. So we feel, and we'll see what referees for this paper think, that this is quite a good measure of female quality, and it relates to throat colour. Okay, what about sexual dichromatism in other Mannophryne species? As I mentioned, Ellen Zaburia's review only mentioned Mannophryne trinitatis. So what we did was we checked the published literature on all 20 of the Mannophryne species so far described, and we also looked ourselves at Mannophryne almani in Tobago. The first conclusion is that the yellow throat colour and the collar are universals in female Mannophryne. This is something that occurs in all members of this genus so far described. When it comes to the males turning black when calling, the other aspect of this colour difference, this has rather rarely been observed or reported. So what we found was it was reported in 8 of the 20 species, and that's including our own observations on almani, and of course lots of people have seen it in Trinitatis. But when you read the papers from the other species, calling behaviour has usually not actually been observed. They've collected the specimens, described the specimens from dead material. They don't call when they're dead, and they don't turn black when they're dead either. So it's uncertain at the moment how many of the species do turn black when the males are calling. But my guess is, if this is a special mechanism that's involved in this genus, it's very unlikely to occur only in a few species and not in the others. So we're predicting in our submission that it will occur in all when people get round to looking at them. Of course, most of the species where it's not been reported are in Venezuela, which is, as you all know, not an easy country to do field work in at the moment. I think it may take some time before this all gets sorted out. So, conclusion, both kinds of sexual dichromatism are likely to be universal in this genus, we think. The yellow throat is a territorial signal of quality, but why do the males turn black? And that's kind of what we're thinking about at the moment. Why should the males turn black when they call? Because the habitat where they live suits very cryptic coloration. It's very, very kind of background rocks and bits of vegetation and dark and light patches, so being cryptic is the thing. Whereas if you turn jet black, that makes you definitely more obvious, especially if you're making a sound at the time. I think the students who've been actually trying to catch these frogs might disagree that being black and calling makes you obvious, because actually they're mostly under rocks and not terribly obvious, but for a potential predator, you'd think it would make them a bit more obvious. I wondered about whether turning jet black might give them some protection as a mimic of other obvious species, because some of the dichromatism, poison frogs, are jet black in part or completely. But for that to work as a kind of mimicry thing, they would have to overlap in their distribution with Mannophryne, and they don't. They're found elsewhere in South America. So that doesn't really work. Our thought at the moment is that it may link to female territoriality. So, since the females are aggressive and maintain territories, and my colleague Paul had a lovely example of female aggressiveness just yesterday when we were looking at some frogs, and this female was there, and the male started calling and approached the female, and the female just jumped on him and chased him away. Females can be really quite aggressive. But one possibility is that the males, in order to attract one of these aggressive females, have to show their quality, and that they are capable of beating off other males. So one way of doing that would be to call, but also to be black and obvious, and to fight another male. And the males do fight each other. So possibly the blackness and the obviousness are something to do with making yourself an obviously good mate for a female. But that's very much a speculative conclusion. One of the aspects of this turning black, though, is that if you're going to make yourself obvious for a short time, you need to be able to turn the blackness off again. And one of the things that we only recently started thinking about is that colour change in amphibians is usually quite slow. I'm sure you're all aware of chameleons and the way that they're able to change colour really quickly. And octopuses change colour fantastically quickly. But in frogs, colour change is usually slow. And it's stimulated by melanocyte stimulating hormone, and it's there to fit you to your background. But just a few years ago, some workers in Australia discovered a frog species where colour change occurs rather rapidly, over minutes rather than days or hours. And they did some experiments on it to see if they could stimulate it to happen using hormones. And what they found was that instead of melanocyte stimulating hormone, this colour change was mediated by adrenaline. So a nervous system stimulus rather than a usual hormonal stimulus. So we think from some observations that Mannophryne trinitatis changes colour quite rapidly. And we're hoping to measure that this year, get enough data on how quickly colour change happens, and we're going to try to see if we can stimulate it with adrenaline. So that's our plan for this year, and well, we'll find out if it works or doesn't. So that's all I've got to say tonight, and we'll take any questions, of course, if you have.